Okay, great. We're very excited today to have Dr. Puneet Rawat joining us. He's a researcher in the Victor Grives Lab in the Department of Immunology, University of Oslo. And we just were chatting offline for a minute, and he was, we were congratulating him on this outstanding paper, and he was saying, yeah, it's been a great team effort, team science effort, culminating after 5 years of collaboration. So always excellent to hear that. A little bit about him. He's a researcher at the University of Oslo, specializing in computational immunology, and his research focuses on structural and repertoire level analyses of B cell and T cell receptors, aiming to advance our understanding of antibody-antigen interactions and immune repertoire diversity. And the title of his talk is Understanding the Role of T cell receptor repertoire in type 1 diabetes status. Thank you again, Puneet, for joining us, and we're really excited to listen to your talk. Oh, I think you went to.
Thank you, Monica, for the kind introduction. And I'll just start with the presentation. So the topic is understanding the role of T cell receptor in the type 1 diabetes status. So it was a very big ask for us to do this project where we are trying to figure out the T1D markers in the PBMCs. So just to start with the brief introduction, how the type 1 diabetes works. So we have, the people have genetic risk, and which causes the beta cell stress. And then you have single autoantibody present in your blood. And then when you have more than one autoantibody, then you are already in stage 1 of type 1 diabetes. And then you become dysglycemic, and then you have the symptoms of type 1 diabetes, where you need the insulin. And then you have a long-standing type 1 diabetes. There are different ways where the type 1 diabetes can happen. It can happen due to the autoantigens or presence of neoantigens, due to the beta cell stress, and also through the this food antigens and the microbial components, different viruses. But coming to our work, so what we have here is that we have sequenced more than 2,000 TCR repertoires, and we have information about the CDR3 beta sequences and the information of the VDJ genes. We also have the patient's age, type 1 diabetes duration, gender, and other informations, along with the HLA information and what autoantibodies they have present. So this is the overall overview of the data set. We have divided our data set into 3 different cohorts, mainly because of the different chemistries we got from the adaptive. So we have to classify different groups. The cohort 1 and cohort 2 and 3 are the different chemistries. But cohort 1 contains all of the clinical groups. We have type 1 diabetic, their first degree relatives, second degree relatives, and also controls. The cohort 2 only has the type 1 diabetes patients, and these are like shallow sequence, same as the cohort 1. But cohort 3 is deep sequenced, as you can see. For the unique clone count, it is deep sequenced, and it only contains the control. So first, what we did is we are trying to see the reproducibility of the results and to see if the shallow sequencing is enough to cover the whole repertoire space. And as we can see, when we compare the shallow and deep sequenced replicates, we see a very good overlap between them. So we thought that we have enough of the unique CDR3 sequences that we can capture the T1D markers, as they have a very low witness rate. Based on that, we defined our 5 main objectives. So we went step by step. So first, we have looked into the repertoire level differences between different clinical groups. Then we also went to figure out if there are public clones that are T1D specific, and to also see how the HLA actually changes your TCR repertoires. And then we have used different classification models using machine learning and deep learning without using any HLA information to find the T1D specific markers. And then we also validated our results on some independent cohorts. So first, coming on the repertoire level differences. So first, we looked into the region uses, and we observed that we don't really see any differences between different clinical groups on their region uses. And same with the similarity and diversity analysis. When we compare the repertoire level similarity based on the hill-based diversity profile, we don't see so diversity. We didn't observe any clustering based on their age, HLA, or diabetes status. And the same goes with public clone and the Mohs-Tahan index. The difference here for the public clone and the Moster-Horn index that public clones does not consider the clonal frequency, where the Moster-Horn considers the clonal frequency. So since we didn't see any difference in the repertoire level, so we thought of like, can we find the T1D specific public clones? And we used two different analyses. And the first one is that we looked into the publicly available database like MCPass and VDGDB. They already have serialized sequences for different diseases, and see if they are present overrepresented in our cohort 1. And for different clinical groups, we observed there are certain diseases that are in which the CDRT sequences are overrepresented in the T1D repertoires. And we also had some sequences from the type 1 diabetes, which was also overrepresented in the T1D cohort. You can see here that we found association with the celiac disease, influenza, HCV, and HIV. So these could be the potential ones that are also associated, like they have some role in type 1 diabetes. To see if there are T1D specific public clones, we did the TCR clustering. So here we have used a bit different protocol. We designed our hypothesis based on cohort 2, where we looked into the TCR sequences that are basically HLA restricted. So how much those sequences are present in certain HLA and in all other repertoires which doesn't contain that HLA. And based on that, we identified those HLA specific CDRT sequences and then tested on cohort 1 to see if they are actually associated with type 1 diabetes or not. We found very few sequences, and most of the sequences were actually protective sequences that are present more in the control and not in the type 1 diabetes. And very few, only 12 TCR features we found that are overrepresented in T1D group, as you can see in the volcano plot. So we found that there are very few public clone sequences that may be T1D associated. So we thought maybe there are not there is no information on the sequence level, but on a subsequence level. So we are trying to figure out motifs that are T1D associated. And to find that, first we looked into the HLA, how it impacts your CDRG sequences. And we know that HLAs are in a very close proximity with the CDI sequences, and you can see with this structure here, it's just a representative structure here, but you can see the antigen is in between, and then there is CDR3 on one side and the HLA on the other side. So there is definitely some impact on the structure level that they are restricting your the frequency of CDR sequences, and that's what we are trying to figure out here. So this is one of the analysis that Ishigaki did in their work, where they have taken the control repertoires and they have figured out how the variations in the HLA can impact your CDR3 frequencies. And the same analysis we did for our own cohort, and we found that the frequencies, the variations actually affects your CDR3 frequencies, as you can see, especially for the DQA1 and DQB1 and the DRB1. They have much higher much lower p values. That means the CDRA sequences, the frequency of CDRA sequences is really being affected by the variation in HLA. So what we have done here to see, so we know there are certain HLA alleles that are present more in the type 1 diabetes, and there was another paper in which they have figured out there are 3 different positions in DQB1 at the site 57, and in the DRB1, the site 13 and 71. So these 3 sites defines around 90% of the phenotypes being the type 1 diabetic. So they have also generated a log odd ratio based on these 3 positions. So we use this log odd ratio, which is called HLA risk score, to see which amino acids are in the CDRT sequences are associated with these HLA risk score. And we did different logistic regression test, and found that there are certain, for each length of CDR3 length of 12 to 18, which amino acids are being restricted by this HLA risk score. And you can see these logo plots here, which shows which amino acids are like positively associated with HLA risk score and negatively associated. And for each regression, we got a coefficient, and that coefficient we called it effect size, that how much it is actually affecting. And using these effect sizes and these amino acids, we have combined them into a CDR3 score for the whole repertoire. And we basically sum up the values for all the effect sizes for each CDR3 sequence, and then calculate the mean over the whole repertoire. And then when we calculate the correlation with the HLD score, we see a pretty good correlation between them. And then to also validate that CDR3 score is good, as a good measure, we also checked, so we have used only type 1 diabetes repertoires and then calculated the CDR3 score for other clinical group as well. And we have done this for all of them as using just using the FDR repertoires or just using control repertoire. Can we see the same trend? And we observe that we always see that T1D has much higher CDR3 score compared to the FDR SDR control. There is always a trend. So once we have these amino acids, the T1D associated amino acids, and we have the effect sizes, we try to figure out, can we obtain a motif out of it? And that's what you see here. So these are different IMGT positions, and for length, CDR3 length of 13 to 16, we found the consensus amino acids that are constantly present and T1D associated. So we, if they are T1D associated, we calculated the PHLA motif score, and if they are negatively correlating, then we also found a protective motif, and we call it NHLA motif. Then we also check these motifs if they really correlate. So do they show association with the different clinical groups? And we see that T1D repertoires has much higher motif score compared to the FDRSM control. And we see the trend, although it's not very visible here, but in the later slide, I'll show that we have a pretty good AUROC value as well. So coming to the next part, so we have used, so this part was HLA association. Now we are trying to classify the repertoires without using any HLA information. And for that, we have used all the previous analysis, the HLA score and the HLA TCR features that are CDR3 score and your HLA motifs as a baseline model. And then we see how these machine learning methods compare to that. So this is your baseline model. So just for our try, we also looked into the conventional high-risk HLA alleles DR3 and DR4, and we see that if one of the high-risk HLA alleles is present, then there is no guarantee that it will be type 1 diabetic. As you see, only the specificity is only 32%. But when both of the high-risk HLA alleles are present, then the specificity increases significantly to 75%. So we also use the HLA score, which is based on 3 different positions, and we see that performance improves a lot. And this is the best performance that we could get, we got in the whole paper. And that's just the HLA score. The only drawback with this HLA score is that it cannot be used as a biomarker for the T1D progression because it's just single value and it's not like time dependent. We also tested the different features that we obtained in the previous analysis. This is the CTR3 risk score on the A and B, and they all have decent AURC values. And then the PHLA and HLA motif also has AURC of more than 0.6 for the cohort 1 and 0.73 for the cohort 2 and 3. And then considering this as a baseline, we use different methods. So first one was to use a machine learning method which uses a public clone. And as we saw that we don't really see public clones for the type 1 diabetes. So the performance was really bad. The high performance you see for the cohort 2 and 3 is mainly because of the deep sequencing of the cohort 3, and which really creates a bias in the result. But you see, for the training, we used cohort 1, and the performance is really bad there. The same happened with the K-mer-based repertoire classification method as well. When we trained our model, we found that the performance of on the training data set is very good. But again, when we tested on the cohort 2 and 3, the AURC goes down to 0.7, which is still decent enough. But we again don't see that there is a trend of the like type 1 diabetes has a much higher motive score compared to the other. It's like the T1D score is much higher compared to all other classes. But when we use the deep learning method called deep RC, here the performance is much better, and you see the trend here that type 1 diabetes has much higher motive the model score compared to the FTR, STR, and control. And the deep RC also has a benefit that we can extract the sequences that has higher weight when it is trying to predict the classes. And we used those sequences to generate a deep RC motif. And in your screen, you can see the motif here. But this motif has two gap positions. So because of that, the motif was present almost like the 17% of the total CDRG sequences in a repertoire were predicted as a type 1 diabetic compared to 0.04% we got from the HLA motif. But still we see the trend here that T1D repertoires has much higher motif score compared to other. And then we also validated these markers on the independent cohort, and we did for all three, but I'm showing only for the deep RC here. So from the GWS analysis, they have identified some risk variants, and we tested all those risk variants for our motif, that do they have higher presence, and we observed that all that you see in A, we checked, and we found that risk variant has much higher motif score. And we also tested different cohorts, the sorted cohorts of spleen and pancreatic lymph node, and we observed that like for the spleen, CD8 has much higher motif score compared to the Tregs and CD8s, and and also we tested for the different in the sorted peripheral blood CD4 T cell subsets, and we found that Tregs has much higher score compared to the other ones. So coming to the conclusion, we didn't see any differences in similarity diversity analysis in the repertoire level similarity diversity analysis. And we found some big public databases, and we found that certain diseases has higher the CDI sequences associated with different diseases, certain diseases has much higher association in the type 1 diabetes repertoires. And we found that there is there are like less public clones in the T1D repertoires, but there are definitely information in the subsequence level. We found that HLA score is the best predictor, but comparable performance can also be obtained without using HLA information and just using the machine learning methods. And all the T1D associated motifs that we found, that they were also validated on independent cohorts. So we found them in the spleen and the pancreatic lymph node as well. And with that, thank you all.
Thank you, Puneet. Thank you very much for this fascinating presentation. I want to open it up for questions. If anyone has a question, please either unmute yourself or drop something in the chat. I had a couple questions. I have to try to keep my eye on this too. You know, when there's a recent paper by Ed Phelps just came out, you know, looking at human slices, live human slices, showing sort of the fragility of beta cells prior to the initiation of the immune response. How does that fit or sort of dovetail with your observations? You know, can you kind of talk about how that might come together with this TCR issue?
Yeah, I think the HLA-based analysis that we have done, we definitely see there is a restriction of CDRG sequences based on the HLA. So the people who are susceptible to type 1 diabetes, even if they don't have immune history, they will certainly have some signatures with them. It's just like at a certain time point, do they have, like, how much signature is present? And
Right.
that will tell about the progression of type 1 diabetes.
Right. And so a lot of, you know, a large majority of islet autoantibody seroconversion peaks around 2 years of age. We've, you know, learned in the, you know, past couple of years. And so how do you think, you know, your results might interface with this situation? How could what you've learned here in terms of the TCR signature be put into play for, detection, monitoring, or, early understanding of what's happening?
Yeah, that's a very good question. Um, so what we have done here is we have used PBMCs. I think once we have like more sorted T cells, then probably we will have better signatures for that. But the signature that we observe in the PBMC is very low, and it's very hard to identify the exact CDRG sequences. So I think that's the main issue right now. And also in our cohort, we have patients from all the age groups. So when we were training like machine learning models, we had to
Mm.
always put some weights, like reduce the impact of those like people with higher ages. So if we have like cohort, which is all the people in the cohort are like 2 years of age at the age period, that
Mhm.
would be much better, I think.
Yeah. So you kind of bin different subjects and really then start to look at them. But of course, it's hard to get the numbers you need, right? The numbers of sample or subjects to really use these deep sequencing methods, I guess, correct?
Yes, yes.
Here's a question from Kagan Crocus, coming out of Aaron Michael's lab. And he's got a question. Have you checked the presented targeted antigens for public TCRs?
I don't think we have antigen information. We haven't checked anything about the antigens, yes.
How do you think your results kind of interface or interplay with any kind of germline defects in TCRs? Could you get a baseline read of any kind of germline phenotype before you start diving into people who actually end up with a biomarker, an autoantibody?
Sorry, I didn't get your question.
I'm just wondering, what do you think how your results might interact with sort of germline defects in TCRs of those who are at risk genetically for type 1 diabetes? Not sure.
I'm not sure, yeah.
That's okay. Anyone else? I see a couple of, I think a couple people from Todd Briscoe's lab are here. City of Hope. Anybody else? Matthias on the West Coast. Any questions? No?
No questions. Just interesting presentation. Thank you.
Thank you.
Thank you.
Yeah, it's a very interesting presentation. Okay, so now I guess the last question is, what are your next steps? What are you guys doing next? Oh, here's another question.
I think the next step would be to have more patients from like the age less than 10 probably. And yeah, and also to have more sorted T cells. So that would be like be much better at finding the TCR biomarkers if we are looking into that. And the paired chains will be, I think, will have more. I think it would be much better to have paired chains, yes, for sure.
And so you guys have access to a large number of samples due to your collaboration with University of Florida, and also you're at at Oslo, you're in Europe. So do you feel confident that you can kind of gain access to these types of samples?
Yes, I think, yeah, we have.
Good. One last question. Any suggestions why type 1 diabetes have less public clonotypes?
I think it's like just the all when you have autoimmunity, then you have like less signatures. If you have like viral infection, you get more signatures for against that. So I think that's the case.
Yeah. You really need to kind of unpack that early window, or maybe just the stages very carefully, and get enough samples at each window right? And really dig into them. That would be really exciting. And it sounds like you're really well equipped now to do that. So congratulations. This is a tremendous amount of work, a 5-year project between really a number of collaborators, but predominantly, you know, Victor Greif and Todd Briscoe. So really some power teams at work there. And thank you, Puneet, for walking us through your work, and congratulations for your great preprint and coming paper.
Thank you. I hope I did justice to this presentation. I was really sick, but I somehow managed it.
I'm sorry, you did a great job. It was excellent. And it was great to see the data and have you walk us through. Have a
Thank
great
you.
rest of your evening and get some well-deserved R&R.
Thank you so much.
Bye-bye now.
Bye.